Hello everyone! Today we are going to talk about the development of a frog. Yes, a frog. Commonly, frogs have protruding eyes, strong webbed hind feet for leaping and swimming, and no tail. They also possess smooth, moist skin. Many are predominantly aquatic, but some live on land, in burrows, or in trees. Besides that, frog is included as an oviparous animals that fertilize their eggs externally. Enough for the introduction, let's move on to the history how frog is used in developmental biology experiment. The frog was historically used by many of the early embryology investigators, and currently there are many different molecular mechanisms concerning development of the frog. The 2012 Nobel Prize in Medicine was awarded to John Gurdon for his 1960s experiment, involving nuclear transplantation with adult nuclei into frog eggs. Other examples include the African cloud frog, which has been used in many embryological studies, as well as the basis of a historic pregnancy test. The leopard frog in 1952 became the first successful nuclear transfer experiment. Enough of history? Now we are going to explain the main part of the video, the life cycle. First is the production of mature haploid gametes, or commonly called as gametogenesis. So, mommy frog will produce ovum or egg, while daddy frog will produce sperm. When the sperm and ovum meet, it is called fertilization. In most species of frog, fertilization is external. In fertilization, gene recombination happens. The genome of haploid male and the genome of haploid female will merge and form diploid zygote. To explain further, the entry of the sperm will facilitate the movement of cytoplasm. This movement is very important to determine the body axis: anterior-posterior or head-tail, dorsal-ventral or back-belly, and right-left. More importantly, fertilization activates molecules needed to begin cell cleavage and gastrulation. In most frog, gametogenesis and fertilization are seasonal events. The male frog grabs the female's back and fertilizes the egg as the female releases them. Some species lay their egg in pond vegetation, then the egg will attach to nearby plants. Other species' eggs float into the center of the pond without any support. Can you imagine when the egg hatches through rapid current? The tadpoles won't survive, so environmental factor holds a huge part on the life cycle of a frog. Now move on to the formation of cell cleavage. During cleavage, the volume of the frog stays the same, but it is divided into tens of thousands of cells, resulted into morula. Morula is a solid ball-like structure containing cells. Next is blastula, same as the previous ball, but it has more cells and it also contains a hollow space in the middle called blastosome. Moving forward to gastrulation. In a nutshell, gastrulation is the formation of gastrula, the three germ layers in the early embryo. Gastrulation in the frog begins at a point across the site of sperm entry, with the formation of blastopore. The blastopore will then mark the future dorsal side of the embryo and expand to become a ring. Cells that migrate through the blastopore to the embryo's interior will become the mesoderm and endoderm. The endoderm is deep inside the embryo and is a precursor of the lining of the gut and respiratory system. The mesoderm is located between the endoderm and ectoderm and is the precursor of connective tissue, muscle, blood, etc. Meanwhile, the cells remaining outside will become the ectoderm and will expand to enclose the entire embryo. The ectoderm is also the precursor of epidermis, brain, and nerves. After gastrulation, the frog went to a stage called organogenesis. This stage begins when the cells of the most dorsal region of the mesoderm condense to form notochord. The notochord produces signals that redirect the fate of the ectodermal cells above it, which will become the nervous system. At this stage, the embryo is called a neurula. The neural precursor cells elongate, stretch, and fold into the embryo, forming the neural tube. Once the neural tube has formed, the epidermal cells and the notochord induce changes in the neighboring regions. The mesodermal tissue adjacent to the neural tube and notochord becomes segmented into somates, the precursors of the frog back muscles, spinal vertebrae, and dermis. The embryo develops a mouth and an anus, and it elongates into the familiar tadpole structure. The neurons make connections to the muscles and to other neurons. The gills form, and the larva is ready to hatch from its egg. The next step in the frog life cycle is metamorphosis. It is when the tadpole will transform into an adult frog through the modification of the organs. The tadpole's tail will metamorphose into the hind limbs and forelimbs for locomotion. The cartilaginous tadpole skull into the bony skull of the adult frog. The lung also enlarges and the gills regresses. Meanwhile, the horny teeth of the tadpole that it used to tear up on plants is transformed as the mouth and jaw takes a new shape, while the intestines shorten to suit the more carnivorous diet of the adult frog. Hormones that induces metamorphosis of the frog is produced in the tadpole thyroid glands. Environmental factors such as temperature and pressure affect the speed of metamorphosis. For example, rana metamorphosis must occur before the ponds freeze during winter. Development of germ cells, such as the sperm and the egg, begins as metamorphosis ends. Gametogenesis takes a long period of time. In rana pipiens, gametogenesis takes 3 years in females for the eggs to mature. And in males, the fertilization of sperm takes a lot less time to mature. For maturation to be completed, germ cells must undergo meiosis and the unity of the sperm and egg cells to restore the diploid chromosome number, which initiates the events that lead to the development and the continuation of the circle of life. Thank you for watching this video. I hope you like it. See you next time!